Okay, so hello everyone and welcome to BrainMap. It's my great pleasure to introduce our speaker today, Dr. Zhongliang Zu. Dr. Zhu is currently an associate professor in Vanderbilt University Medical Center. He received his PhD degree in medical physics from Peking University in 2008. And after that, he joined Vanderbilt University Institute of Imaging Science as a postdoc fellow and became a faculty member in 2012. His research focuses primarily on the development of advanced MR sequences, including chemical exchange saturation transfer or CEST, magnetization transfer, spin lock, and more for molecular and physiological imaging, and their application in tumor, ischemic stroke, neurological, and muscular diseases. I would just like to remind the audience to please address any question you have using the Q&A box. Professor Zhu, thank you very much for coming here today. The virtual stage is yours.
Okay, thank you for our introduction, and thanks for inviting me to give this talk. Thanks for everyone who participated in this seminar. Okay, so today I will introduce specific CEST MR imaging and new NOE signal at around -1.6 ppm. Okay, so in 2000, Walden and Bilibin has suggested using exchangeable proteins for MR contrast. They have demonstrated the possibility to turn on and off this contrast by selective separation of protein interest, and named this approach as chemical exchange separation transfer, or CEST. After that, many interesting CEST molecular imaging contrasts were reported, such as APT, amide protein transfer, which can measure the protein and peptide content and intracellular pH. And GAG-CEST can measure GAG, GLUCEST can measure glutamate, glucose-CEST can measure administrated glucose, and creatine-CEST can measure the creatine and phosphocreatine. And acido-CEST can measure extracellular pH, and urea-CEST can measure urea. Because of this intrinsic contrast mechanism, they have been applied in many clinical applications, such as the APT imaging of protein and peptide can grade tumor, can distinguish active glioma from treatment effect. APT imaging of pH can identify ischemic P number, differentiate ischemia from hemorrhage. And the GLUCEST can diagnose neurological disorders, and urea-CEST can detect the kidney function. And creatine-CEST can detect the creatine kinases in the muscular diseases and cardiovascular diseases. However, although CEST show potentials, it has a low specificity. So here is a solute pool. We know that direct measurement of the solute has low sensitivity because of its low concentration in our body, or because of the short T2 of the solute. Here's a water pool, which is abundant in our body. By measuring the change in your water signal due to the chemical exchange effect between the saturated solute protein and the water protein, we can indirectly measure the solute with enhanced detection sensitivity. This is the interest of CEST. However, water signal also depends on T1. So this raises the first question: do we need to correct T1? This issue is still under debate. Some people think that we need to correct T1 based on the fact that water is influenced by T1. But some people don't think so, because they found that some interesting contrast in tumor disappear after correcting T1. So following, I will introduce my suggestion about this question. Besides solute, there's also MT effect between the semi-solid pool and the water pool. So this can also change water signal and raise the second question: how to isolate CEST from MT and other non-specific factors? So this is the active research field. Until now, there are many approaches have been developed to address these issues. So following, I will introduce my approach. There are also overlapping solutes. For example, creatine has amine at 2 ppm, but protein also has arginine amine at 2 ppm. Their exchange rates are close, and thus it's difficult to separate them. So this raises the third question: how much is the contribution from overlapping solutes? So this issue has not been noticed until our publication a few years back. Following, I'll introduce how I analyze the contribution from overlapping solutes. Okay, now the first question, do we need to correct water T1? I will analyze this issue by developing a signal model, which shows the dependence of CEST signal on T1. So here we have a three-pore exchange model. We use W to represent water, S to represent solute, and M to represent semi-solid pore. We have MZW, MZS, MZM represent the longitudinal magnetization of the three pore respectively. And we also have eta, delta, and alpha representing the acceleration or labeling efficiency of the RF pulses on these three pores respectively. And we also have KSW and KWM, which represent the chemical exchange from the water to solute and from water to semi-solid pore respectively. Okay, in CEST imaging, the water magnetization loss due to chemical exchange effect could roughly be the product of MZW, KWS, and delta. The water magnetization loss due to MT effect could roughly be the product of MZW, KWM, and MZM. And the water magnetization loss due to DR wall acceleration could roughly be the product of MZW and eta. So the total water magnetization loss could roughly be the sum of these three terms. Besides magnetization loss, there are also water magnetization recovery, which is due to the R1 relaxation and can be this formula. And MZ can be, in steady state, the two formula are same. So the water magnetization loss is equal to the magnetization recovery. So MZ can be derived from these equations to here. Actually, so MZ, the water magnetization can be obtained from a spin lock theory. So here I use simple derivation to get this equation. So the simple derivation can allow us to have intuitive understanding of why the CEST signal depend on the non-specific factors. Okay, so asymmetry analysis is a widely used CEST method to quantify CEST effect, especially in the clinical human study. The asymmetry analysis is termed MTR asym. The study's magnetization can be expanded to Taylor series in power of the chemical exchange effect. So here we have the zero order term and the first order term. This can also represent the label signal. And in reference signal, the exchange effect is zero, so KWS is zero, and we can get the reference signal by setting KWS to zero from this equation, and we get this equation. And MTR asym will be the subtraction of the label and reference scan and normalized by the control signal. And then we get this formula. Compare this formula with the label signal. We can find that the MTR asym can only remove the zero order term, but not the first order term. So we can find that the MTR asym still depend on MT and acceleration effect, and also has complex dependence on R1W. Here we ignore the MT effect and reform this equation to this formula. And please note this term and compare this with the reference scan and ignore the MT effect. So we can find that this is actually the water signal with off resonance RF acceleration in a single water pool model. And this can be obtained from the block equation. And then this formula becomes this formula. So here we have omega-1, which is acceleration power, delta omega, which is the acceleration frequency offset. So this term is related to acceleration effect. And so here we have R1. It influences MTR asymmetry through the acceleration effect. So we term it as R1 related acceleration factor. In weak acceleration effect, So this term will be 1. And then the MTR SM will only depend on this R1. And so here, since there's no saturation effect, and then there will be only recovery. So we term this factor as R1 recovery term. So now we have 2 factors and 2 R1, and we can find that the 2 R1 has inverse influence on MTR SM. So it's probably that we vary we adjust omega 1 so that the MTR SM is roughly independent on R1. Okay, so here's our simulation. And we have a 2-pore model simulation. The 2-pore contain water and a solute. And this is a 2-pore model simulated MTR SM as a function of T1. In the upper figure, we mimic the weak saturation effect, in which the RF offset is 10 times of the RF saturation power. In the lower figure, we mimic the strong saturation effect, in which the RF offset is 5 times the saturation power. So we simulate several powers from 1 to 5 microtesla, but keep this equation constant. And the different power are represented by different color. Okay, so let's see what we find. So from the first figure, we can find that the MTR asymmetry is roughly linearly depend on T1W. But the lower figure shows that the MTR SM is roughly insensitive to T1W. So this suggests that at high field, where the saturation is usually weak, we need to correct T1. But at low field, where the saturation effect is strong, it may not be necessary to correct T1. So I think this may partly explain the current debate. Okay, besides T1, there's also MT effect. So here, so we also perform a 3-pore model simulation to study the influence from MT. So here the 3-pore model contains the water, solute, and the semi-solid pore. And we have 3-pore model simulated MTR SM as function of FM. Here the FM is the semi-solid pore concentration. So also the upper figure shows the weak saturation effect, and lower figure mimics the strong saturation effect. We can find that in both of these situations, the MTR have strong dependence on the MT effect. Okay, so in human clinical imaging, non-steady state acquisition were usually performed. So this is because of the limitation of the SAR or the hardware requirement. So the non-steady state magnetization decay with following exponential function and reach steady state magnetization. So the decay rate can be described by R1 rho, which can be obtained from a spin lock theory and equal to the sum of R1, exchange MT, and saturation effect. I will not explain the spin lock theory here since that's complex, but we can get that from, we can derive this equation from the spin lock theory. And also, the steady state MZ can be also obtained from the spin lock theory and equal to the ratio of R1W and R1 rho. And here, we expand this exponential function to Taylor series in power of R1 rho. And then we input these factors back to this equation. And then we get this formula. This can also represent the label sequence. And also in the reference signal, the KWS is 0, and we can derive this formula from this equation. And also the MTR SM will be the subtraction of label and reference. And then we can get this formula. So note that the non-steady state MTR SM only depends on the CEST effect and the saturation time, but not the MT and T1. So here is our simulation. So the upper figure shows the 2-pore model simulated MTR as a function of T1, and lower figure shows the 3-pore model simulated MTR SM as a function of FM. Our simulation, the saturation time is 0.5 seconds. which is usually used in the non-steady state acquisition. And also we mimic the strong saturation effect in the upper and the lower figures. Okay, so in the upper figure, we can find that the non-steady state MTR asim is roughly insensitive to T1. And in the lower figure, we find that non-steady MTR has some dependence on MT, but compared with the steady state MTR as a function of MT, so this dependence is relatively weaker. Okay, so in this small this subsection, I introduce a signal model guided by which we can know that how the set signal depend on the T1. So take-home messages at high field, such as 9.4 Tesla, it's necessary to correct T1. But at low field, such as 3T, or with non-steady state acquisition, it may not be necessary to correct T1 when using appropriate sequence parameters. Okay, now the second question, how to isolate sets from MT and other non-specific factors. So usually the ideal model has a symmetric MT effect and only one solid pool. But in real biology tissues, the MT is asymmetric and there's many exchange or dipolar interactions pools. Here is a Z spectrum we acquired on red brain at 9.4 Tesla. Note that there is some multiple dips, and we have used multiple landing fit to resolve each peaks. So here you can find the fitted amine, NOE, water pool, and the asymmetric MT. So it looks this work well, but take a very long time since you need to acquire the whole Z spectrum. This is not impractical in some applications, such as stroke, where we need to rapid imaging, and in some situation where patients are weak. So to address this issue, we have developed a method termed chemical exchange rotation transfer, CERT. So this is a fast approach to isolate slow exchanging pools. So this is CERT sequence. It contains two pulse sequences. So the first sequence, we have a series 180 pulse followed by data acquisition. In the second sequence, we have a series 360 pulse followed by data acquisition. We vary the delay time or some other parameters so that the two sequences have the same average acceleration power. Okay, now let's see what's happening when we perform the sequences. Before that, let me introduce the several spins which represent the major tissue component. So here, the first spin represents the slow to intermediate exchange. This includes the amide, which has a exchange rate less than 100 Hertz and belong to the slow exchange region. And it also includes the arginine amine, which have an exchange rate of several hundred Hertz, which is belong to the intermediate exchange region. And this spin represents the semi-solid pool. And this spin represents the dear water acceleration. And this spin represents fast changing pool, which include the glutamate amines or glucose hydroxyls, which has exchange rate of several thousand Hertz, and they belong to the fast change region. Now let's perform a pulse sequence with 90 degree. We can find that only this spin rotates to nearly 90 degree, while other spins only shrink to a level. Well, when we perform the pulse sequence with 180 flip angle, but keep the constant flip average power at that with the flip angle 90, we find that only this spin rotate, but the other were kept constant. And the 360, why? So first, this is because that for these spins, the RF pulse is applied on solute. So it's actually on-resonance excitation, so it has rotation effect. But for the semi-solid pool, since the solid, the pool T2 is very short, and so we don't have rotation effect. And for direct wall acceleration, our RF is applied on solute, but not water. So this is actually off-resonance excitation. So the spin will rotate around the effective field. So there's also no actual rotation. And for fast change, during the flip angle, during the acceleration pulse or rotation pulse, the spin will exchange. And so there's also no rotation. So this becomes less effective rotation. Second, so we can find these three pulls does not depend on the flip angle, but they depend on acceleration power. What the acceleration power is kept constant. Whatever the flip angle is, their level is the same. So this interesting spin feathers transfer to water and cause interesting response from water signal. So here is our measurement on rat brain by using the pi-iteration. So we can find the deep to deep. This is amide at 3.5 ppm, and this is arginine amine at 2 ppm. And we also acquire 2 pi. So you can find that the curve from the 2 pi isolates the background. Why? This is because when we use the pi irradiation, the spin will inverted. And then after transfer, it will cause maximum of CEST effect. But when you use 2 pi, the spin will rotate it back. So after exchange, it will cause minimum CEST effect. But for both of these sequences, they have the same edge power, and thus MT direct acceleration are the same. And so they have the same this effect. So we subject this two curve, and we can get the third spectrum, and we can remove the MT or direct acceleration and can isolate the CEST effect. So here we have amide and arginine amine. And there looks a dip here, which may be from phosphorylating. Okay, so here's MTR-ASIM, a conventional asymmetric analysis. We can also see the amide and the arginine amine, but they are overlapping on the sloping baseline, and also their value are negative. So the conventional asymmetric analysis can only provide a weighted signal, while our third method can quantify the signal. Also compare with the multiple learning fit, the search is fast, since we don't need to acquire the whole CT spectrum, and 2 points at the CEST effect will be enough for quantify the CEST effect. So it's very fast. So here is our experiments on rat brain by bearing Nile tumor. So here's a third map. This is a conventional MTR-ASIM map. So here you can find that in the normal tissue, the value is negative. So this can only provide the weighted signal. And recently, our collaborators have applied this method to 3T MRI on a human brain. The image looks good, but we are still optimizing it, since when the RF is very close to water, and the effective field will close to the transverse plane, and thus there also will be some rotation effect. And then the third will has contaminations from water-derived acceleration effect. But we're still optimizing it. Besides the third, there are also many other approaches, such as unresonant paramagnetic chemical exchange effect, flex, sulfury, lava, VDMP, and CEST. And all this method imply the different feather of the spins and to isolate the CEST effect. However, currently, most methods are still apply on the high field preclinical scanner. So I think in future study, it's important to translate them to 3T MI. Okay, so take-home messages. The resonant frequency of tissue components are close, but their exchange rate or relaxation are much different. Designing sequences that are sensitive to exchange rate and relaxation may be a better way to isolate CEST from non-specific factors. Okay, so the third question, how much is contributions from overlapping solutes? Now let me first introduce the background. We know that creatine has amine at 2 ppm and glutamate has amine at 3 ppm. But protein also has arginine amine at 2 ppm and lithium amine at 3 ppm. Here's our phantom study to evaluate the contrast region of CEST signal. Here we have the major tissue components, including the creatine, EWA, which is a protein from egg white, and glucose, glutamate, myo-inositol, and phosphocreatine. They are in their physiological concentration and pH, and we measure those phantoms at 37 degrees. Here, the red line represented the creatine, while the dotted red line represented the protein. Other colors represent the other metabolites. So you can find that the CEST signal at 2 ppm from the creatine dominate all other metabolites, but the CEST signal at 2 ppm from protein is comparable to that from creatine. So this cannot be ignored. Here we also evaluate the contrast, the origin of the CEST signal. We also measure these major tissue components. And here we use a higher power, 3.6 microtesla. So this power has been used in the first CEST paper published in Nature Medicine. So here we found that the blue line is a glutamate. Dotted blue line is from protein, but that signal is higher and beyond this image. So here we can find that the CEST signal from the glutamate dominate other metabolites. But in this figure, we scale the y-axis so that we can see the dotted blue line, the protein. We can find that the signal from protein dominates all metabolites, including the glutamate. So this result suggests that CEST signal from proteins cannot be ignored in CEST imaging of metabolites. However, current validation of CEST metabolite imaging was only performed on phantoms containing major tissue metabolites, but ignored the contribution from proteins. Why? I think this may be because that there are 100,000 type of proteins which have a different proportion of these residues in our body. So it's difficult to mimic them using simple protein model phantoms. So that's why I think that our previous validation ignored the contribution from proteins. Well, I address this issue by using Daly's tissue homogeneous. We know that tissue homogeneous contain nearly all the metabolites and the proteins in our body. And Daly's tissue homogeneous with Daly's membrane with a molecular cutoff for like 1 kDa can remove the metabolites from the proteins. So here we put the Daly's bag in a container contain PBS buffer, and this will stay there for a few days, and we change the PBS for a few times, and then you can find that the metabolites will diffuse out. So we think this is a good model to evaluate the relative contribution from proteins and metabolites. And we have evaluated the molecular origin of a CEST using this model. So here we have measured the MRS on this dialysed tissue homogeneous, the red line, and the control tissue homogeneous, the blue line. So we can find that after dialysis, the creatine signal decreased greatly. This suggests that the successful dialysis of this method. And this is a CEST experiment, the Z spectrum on the dialysed tissue homogeneous, the red line, and the control tissue homogeneous, the blue line. We can find that at 2 ppm, the CEST signal decreased greatly after dialysis. So this change could be due to the creatine. And we have quantified this effect by displaying interpolation of the background signal. And then we get this quantified CEST signal. So we found that maximum 66% of CEST signal at 2 ppm in brain may be from creatine. We have also evaluated the contrast origin of a glucose signal. So here's also the MR study. We found the glutamate signal, MR signal decreased greatly. And here is, excuse me, CEST signal, this spectrum on the dilute tissue homogeneous and control tissue homogeneous. The red line represents the dilute tissue homogeneous, and we measure it with 3.6 microtesla. We found that the dilute tissue homogeneous decreased greatly on both sides of the water peak. Why it disappears on both sides of the water? This is because that at higher power, the CEST effect is mainly contributed from the fast changing, and the fast changing pool have broader CEST effect, and so they extend to the arc field. And okay, so here we use asymmetric analysis together with correction of sloping baseline to quantify the glucose signal. The asymmetric analysis has been used in all current glucose imaging, but they did not correct the sloping baseline. On 9.4 Tesla, we found that without correcting baseline, the curve will be below 0. So we corrected this baseline, and then we quantify the signal. And we found that maximum 24% of CEST signal at 2-3 ppm in brain may be from glutamate. This is low, but we think this is still okay for glutamate imaging, since the first glutamate paper reported that the glucose has 700 signal enhancement than RMIs. So 1/4 of the CEST signal from a glutamate is still okay. And we also found that the asymmetric analysis with the suppression of signal at 3 and -3 ppm may reduce some CEST signal from glutamate. So if we can measure, quantify the glucose signal at 3 ppm by removing the baseline with fitting or some other approaches without using the asymmetric analysis, probably we can enhance the detection sensitivity. Okay, so here we use the dilute tissue homogeneous to evaluate the contribution from proteins to current CEST molecular imaging. The method looks not perfect, but for example, the phosphocreatine can become a creatine after postmortem, but this can be reduced. This reaction can be reduced if we can quickly freezing the tissue, and can use some other chemicals to reduce this reaction. Also, there's some restricted metabolites that cannot be diluted out, but their concentration is low. So whatever our method can provide a rough estimation of the relative contributions from proteins and from proteins and the metabolites. Okay, so take-home messages. Here in specific tissues, the molecular region of CEST effect could be evaluated by dilute tissue homogeneous together with phantom containing major metabolites. Second, interpretation of glucose contrast in the disorder should consider the contribution from variation in proteins. Okay, so in the above part, I introduce 3 issues that are related to the CEST specific imaging. In the first issue, that is how much do we need to correct T1? So I suggest that it's necessary at high field, but it may not be necessary at low field or with the non-steady state acquisition. So this may address current debate. Second, how can we address remove the MT and other non-specific factors? So there are many approaches now, but I think the next step is important to translate them to 3T MRI. And the third issue is that we found that protein has great contribution to CEST molecular imaging. So interpretation of CEST imaging metabolites should be careful. Okay, so in the second part of this talk, I want to introduce new NOE signal at around -1.6 ppm. So here's this spectrum. It's on a red brain at 9.4 Tesla. And we have 98% oxygen for maintenance. So on this spectrum, we can find there's multiple dips. This is an amine and NOE -3.5 ppm. And this dips has been reported widely previously in many publications. But it looks there's a dip here, but it's very close to 0 ppm, and it's hard to say, is that real? Based on my experience, many people don't think it's a real signal. But until recently, we have injected some chemicals in the rat, and then measure this spectrum again, and we get this spectrum. So you can find that the change signal, or the change is here between -1 to -2 ppm. So I think you must be want to know what's going on here. It may be a real, so I think some people may think so. So in following part, I will introduce what I found about the variation of this signal in several pathologies and disorders, and my preliminary study on the molecular agent, my hypothesis, and the future plan. I named this signal as NOE -1.6. So first, I found that NOE -1.6 can be enhanced with the intake of oxygen. So the first figure is quantified NOE -1.6 for animals with intake oxygen and air. This is created from 10 animals, and we can find that there's a significant difference. And the second figure shows the quantified NOE for each animal. So here we can find that with intake of oxygen, the signal can be enhanced for all the animals. We also find that the variation of the NOE signal with intake of air has a bigger standard deviation than that with oxygen. So we also performed the third plot. So here is a change of NOE after intake of oxygen as a function of NOE with the intake of air. You can find that when the NOE with the intake of air is high, the enhancement is not is small, like the number 9 and 10 are red. When the NOE with intake of air is weak, the enhancement is big. So this may show the saturation effect of this dip. Currently we are still not known as a mechanism. We're still working on that, but the enhancement with the oxygen, I think that may be the reason why we first report this signal. So I have asked people in several institutes, and I know that in their preclinical study, they use air for maintenance. But in our institute, we usually use oxygen. So I think that's why we found that the clear dip. Okay, second, NOE decrease in ischemic stroke. So here's a rat with a stroke model. We have the fitted NOE -1.6, fitted APT signal, NOE -3.5, and amine. And this is the baseline. And this is a half hour to 1 hour after onset of stroke. And this is a 1 to 1.5 hour and 1.5 to 2 hours. And the lesion is on the left side of the rat brain. So we can find that just after onset of the stroke, the lesion become dark, and this also for APT become dark. And we also performed this is statistical result with 6 animals. So NOE, AMITE, NOE -3.5, and amine. We can find that the NOE -1.6 show better significance, and also the contrast is bigger than others. Here we still don't know the contrast mechanism, but I'm sure that this is different from the APT. We know that the APT detected the decreased pH in stroke, and we also know that the NOE could be direct dipole induction, and could also be the chemical change related dipolar interaction. If it's related, it should depend on both the chemical change and the dipolar interactions. But usually the dipolar interactions has lower rate than the chemical change. So the final effect should depend on slower rate, but not the faster rate. So we think that the NOE should be a new contrast. It should not be related to the pH. Okay, so 1 year after our publication, another institute also repeated our study. And so this is a NOE. They also find good bigger contrast. And this is their maps. The last one is NOE minus 1.6. So you can see the lesion clearly. We also find the NOE decrease in tumor. So here is the NOE minus 1.6 map, the NOE minus 0.5 map, and R1 map, and the empty post-size ratio map. So here's the tumor. And this is a statistical result from 8 animals. This is NOE minus 1.6 and minus 1.5 and R1 and the empty post-size ratio. And you can find that the contrast is bigger than others. And this variation in tumor can be also found in a previous publication published in 2013. In this publication, they studied the NOE in a human brain at the 70, and this is this spectrum and the fetus spectrum from a healthy tissue, and this is from a tumor. Here you can find that in the healthy tissue there's dip. So this is minus 1.6, minus 5 ppm. So this should be minus 1.6. So you can find this dip disappear in the tumor. But in this publication, they only report in the study on the NOE minus 3.5. The NOE at minus 1.6 were not noticed in this publication. Also, we found that NOE decreased after postmortem. So here is this spectrum. The blue line is from the lab animals. The red line is from after postmortem. So you can find there the dip become more smooth here. Also here is the statistical result with 5 animals. So you can find that after postmortem, they decrease significantly. So this variation of the NOE in pathology and disorder suggests that the contrast region may be interesting. So I performed preliminary study on this region. So here's some MRS spectrum. We know that at minus 1.6 ppm, there could be contribution from taurine, choline, and creatine. But we know that NOE should be from micromolecular or immobile metabolized. But creatine, choline, and choline-containing metabolites and taurine are in free state, so they cannot be the origin. But choline phospholipid account for 60% of total membrane lipid. So this figure shows the molecular structure of the choline phospholipid. So we have the choline head group and connected to the lipid chain, two lipid chain. The lipid chain is inserted in the membrane, and thus the choline head group is in a restricted state, which may allow the dipole interactions between the choline head group and water protons. So we have that the NOE may be from the choline head group. And then we performed a study on a phantom. This is reconstituted membrane phospholipid made of HPC. And we found the dip at around minus 1.6 ppm. And we also found the dip at minus 3.5 ppm. So, but some people may say that this may be a direct measurement of the choline lipid, but not through NOE. So we also perform additional studies. So here is this spectrum we measured with a field time of echo, and we measure using FID acquisition and using the first point to create this spectrum. And we then get this plot. They show the quantified NOE signal as a function of time of echo. So if the signal is if the NOE signal is from the direct measurement of choline lipids, the in-phase and out-of-phase of the choline head group and the water speeds will cause oscillation of the signal. If that is from NOE, so there should no oscillation. So here we found there's oscillation, and we have measured the cycling here. That is 1.5 millisecond, and it's also roughly to 1.6 millisecond. And we have calculated this is in phase and out of phase of cycling at 9.4 Tesla for the choline head group. So it looks this is a direct measurement, but compared with this part, this is small. So we think that the NOE should be mostly from the NOE effect, but not the direct measurement. And so here, the NOE has 4.5 times signal enhancement. So you can find here the dip is narrower than that we get on animals. We think that in membrane, the membrane is more rigid, and thus the choline head group T2 is more shorter, and thus the dip is wider, and the T2 is shorter. So in that, we think that in biological tissues, the enhancement should be higher. Also, I think most importantly that the NOE cannot only measure the choline head group concentration, but the coupling rate, which we think should be related to physiological conditions. This is similar to the APT imaging of the protein peptide content and pH. Okay, so we have found that the signal change with intake oxygen. So this is possible that the signal is related to blood. So we have performed additional study to suppress blood. So the first one is we're using high p-value using diffusion weighting. So this is the Z spectrum required with no diffusion weighting and with high p-value. And this is a fitted peaks. We have AMIT, NOE, and NOE minus 3.5 ppm. And we have the statistical result from the AMIT, NOE minus 1.6, and NOE minus 3.5 ppm. And confirm that with high p-value, the signal does not change significantly. This suggests that the NOE signal should not be mainly from blood. Also, we measured the Z spectrum on ex vivo blood, and we found that the NOE signal is relatively smaller than that measured in the live rat brain. So this two studies suggest that it could not be from blood. So we also performed further study to suppress blood, the Mayang. Interestingly, most surprisingly, we found that after injection Mayang, we found the signal, the red line is after injection Mayang, decreased significantly at minus 1.6 ppm. So this is a figure that I show in the first page of this part. So this is a fitted peak, and this is statistical result from AMIT, NOE minus 1.6 and NOE minus 3.5. We found that this NOE minus 1.6 decreased significantly. So we still don't know the mechanism, but if this two studies suggests it's not from blood, but the Mayang cannot cross BBB and thus stay in the vessel. So the flowing Mayang in the vessel, the only way it influences the NOE should be it changed the electromagnetic environment in the brain parenchyma. So that's our guess. And we also here I want to mention that the NOE minus 3.5 ppm signal does not change. This may be that we guess that this is from choline head group, but the NOE at minus 3.5 maybe from the lipid chain, and their dynamics is different, and so maybe this is happened to in the frequency range of the electromagnetic the change in the electromagnetic that induced by the Mayang. So here is our hypothesis. The phantom study suggests that NOE maybe from dipole interactions between phospholipid choline head group protons and water protons. Second, the Mayang study may suggest that the Mayang particle in flowing blood at a fluctuation field to brain parenchyma, which may reduce the dipole spectral density close to zero frequency. This is equivalent to the change of correlation time and thus decrease of the NOE effect at minus 1.6 ppm. Okay, so in this study about the NOE, we found that NOE vary in stroke, tumor, gas challenge, and after postmortem. And the NOE molecular origin may be from the choline head group. But it looks what we found is just the top of iceberg. So a lot of things that we need to discover, like the contrast region in stroke. Could that be due to the pH or the variation of the blood? And the contrast region in tumor, could that be due to the change the content of choline lipids or the vasculature? And NOE region in gas challenge, could that be due to that the oxygen change the chemical, the electromagnetic environment, and whether it changes in other pathology and disorders, we don't know. For many years, I don't have any progress on these questions until recently. I inject Maya and find and get progress. So by adjusting the dose of Maya, we can avoid the change of T2, but can have a significant change of an NOE signal. So this can suggest the presence of the NOE signal and the variation of the signal in different pathologies and disorders. So here I think the Maya will be a very good tool to study this new signal. So now I'm confident for this signal is real, and we have a good study tool. So I want to encourage everybody, if you are interested in this signal, just try to check whether it's present in your study. Just injection Maya. So finally, probably we can get large data from various pathology and disorders. We can finally analyze the molecular origin. Okay, hopefully we can play beautiful music at -1.6 ppm. So I want to thanks for our collaborators. Xiaoyong and Jing is my postdoc. They have performed measurement and data analysis. And here I want to also thanks Taojin, who is from Pittsburgh. And there's a story here. About 10 years ago, I sent this spectrum to Taojin and asked him a question. It's not related to NOE, but Taojin replied to me. He found that he told me that it's very interesting dip at -1.6 ppm. So after that, I came to notice this dip and pay attention to it. So the first one who found this signal should be Taojin. Also, I want to thank Feng Junzhong, Jinping, and Goldberg and Dr. Gore for their helpful suggestions and help on my experiments. Okay, so also thanks for the funding agency. So all my funding is related to validation and the specific imaging of the molecular engine, including NOE, creatine, and glutamate. And also thank you very much for your attention.